Welcome again to the Southern IPM Hour. We're going to talk about thermotherapy and strawberries today. And this is a series presented by the Southern IPM Center, where we present research issues and programs in integrated pest management from the Southern region through this webinar series. So the IPM Hour usually takes place on the first Wednesday of the month at 1 p.m. Eastern, although this month we have several that are coming up, so we're really excited about that. Starting with this talk today. And I'm Kayla Watson. I'm the Communication Director of the Southern IPM Center, housed at NC State and the University of Georgia. And we have a mission to coordinate IPM across the region. We are one of 4 regional IPM centers supported by USDA NIFA Crop Protection and Pest Management Regional Coordination Program. So a couple of announcements from the Southern IPM Center. Next week, we're going to release our call for proposals for our annual Southern IPM Grants program. So that's always exciting. We have up to $30,000 for projects and up to $40,000 for working groups, and that's for each individual project. Usually it caps out at a total of $250,000, about 12 projects in total. So be on the lookout for more information about that. And you can see general information on our website now, as well as past projects, so if you want to check that out. And we're also accepting nominations for our Friends of IPM Awards program. And that's to celebrate those who have done really outstanding work in IPM in the Southern region, either as a student or professionally. There are 7 professional categories and 2 grad student categories. And I'll put links to both of those programs in our chat. So the Friends of IPM program and then the annual Southern IPM Grants program will open up next week. But for the webinar today, if you have any questions for the speaker as we go throughout the talk, we'll be answering those questions at the end. So you can type those questions into the Q&A located on your Zoom control panel. And our speaker today is Dr. Natalia Perez. She's going to be speaking with us about her ARDP project, and that stands for the Applied Research and Development Program under the Crop Protection and Pest Management Program, NIFA. And this project looked at using thermotherapy in strawberries. And Dr. Perez leads the Perez Plant Pathology Lab at the Gulf Coast Research and Education Center at the University of Florida. And they conduct basic and applied research on important diseases affecting strawberry production in Florida, among other things. So welcome, Natalia, and we're looking forward to your talk today.
All right, thank you, Kayla, for the introduction, for the invitation to be here today. Thank you, Roger, as well. So as Kayla mentioned, I'm at the University of Florida, based at the Gulf Coast Research and Education Center that's just located just south of Tampa. So I would like to start my presentation with the acknowledgements now that you're awake, rather than at the end when we might be sleeping. The project I'm going to talk specifically today, or actually go into more details, the CPPM project. But I have started working thermotherapy a while back, and I have collaborated with several people in this during this time and had funding from different agencies and programs. So, and I would like to acknowledge the people I work with, in particular, Bill Turshak. We started working together back in 2007, I believe. And over the years, several other people. For the CPPM project in particular, I'm the PI, and Bill and Johan Deseger, who's a nematologist, our co-PIs. Other collaborators over the years include Ole Marine and Arne Stensven from Norway, several students and postdocs in my group, Nani Wang, Adrian Zuniga, Juliana Baggio, Marcos Marine, Teresa Sejo, Robert Marking, as well as Justin Reckma. Justin used to be a faculty here at the UF. He's now in Canada. And several growers that we have collaborated over the years, in particularly nurseries that we have done extensive trials, Lassen Canyon, Blue Nursery in Quebec, Northern Creek and Aston Nurseries in North Carolina, as well as Trisco's in California. As far as funding, we have the CPPM, as I mentioned, but we have also got previous funding from SCRR and initially from IPM CAR program, as well as from our Growers Association for the Strawberry Growers Association, FSGA. So I'd like to start with some background and justification. First, a little background about strawberries in Florida, which might be different than some of the people in the audience that grow strawberries up north. But I think it doesn't matter where you grow, a strawberry is a high value crop and also a high risk crop. And here I just have some numbers for Florida to give you an idea of the value of the crop and the amount of investment that goes in. So it generates over 300 million a year, but it's also a high investment where it costs in Florida about $30,000 to grow an acre of strawberries. I know it's even higher in California. And considering the average size of about 30 acres and that we have in Florida, you can quickly do the math and see how much investment it goes in to grow strawberries. One of the uniqueness of Florida is that most of the production is located in one county, which is Hillsborough County. That's where 95% of commercial production is concentrated in Florida. And that's basically pretty much where our center is located. We are within 20-30 mile ratios of most of our growers, which gives us the ability to work really close to them. So, in Florida, we grow strawberries as an annual crop, which again might be different in some of the other places in the north. We're about to start our season. We plant in the end of September, beginning of October, and the season goes all the way to March, April. It really depends on the market and in competition with California and Mexico. And at that time, we kill the crop and then we'll wait to replant the following season. But one of the key aspects of production that I want to talk about is this transplant production. Those the transplants that we grow in Florida are what we call green top, bare root. They come with leaves, as you can see, and they're bare root, as I just said. Those transplants, though, they're not produced in Florida. They're produced, in fact, far from Florida by different growers, not the same growers that will grow the plants here, grow the transplants. Most of the plants that come to Florida are produced in nurseries, either in California, North Carolina, or Canada, and there are 3 different production areas in Canada. Those transplants, they're certified disease-free, but the certification is really and only for systemic diseases like virus and phytoplasma. So their initial transplants are grown in tissue culture. They are propagated in screen houses and greenhouses where they are limited with the exposure to insects is limited. But after the first testing that they're clean of virus, they are multiplied in open fields, as you see in the pictures, where you plant the mother plants and you let those plants produce runners, and the number of runners will depend on the cultivar and the area you're producing. But at the end, when you're ready to harvest those nurseries, you have fields like you see at the bottom here. The multiplication in open fields, it depends on what level of plants you have. It can go for 2, 3, sometimes up to 4 generations before you get the plants in Florida. So with that and the fact that you're growing in open fields, it just brings a lot of chances for those plants to be exposed to pathogens. So those plants are considered high risk for our growers because you often don't know which one of those plants will be coming infected with any of those pathogens that you see here. So I broke down here the most important pathogens as fungal and bacterial. There's only one important bacterial disease and fact for our region, which is bacterial leaf spot or angular leaf spot caused by Xanthomonas regari. Most of the other diseases are fungal diseases. On the top here, I have crown rot diseases caused by Phytophthora, mostly Phytophthora cactorum, Coleototricon, mostly species within the Coleototricon Goisporialis complex, as well as Macrophomina phaseolina. And the fruit and foliar diseases like botrytis fruit rot caused by Botrytis cinerea, anthracnose fruit rot caused by Coleototricon maculatum, powder mildew, and more recently we're having issues with Pistologia leaf spot and fruit rot caused by what we think might be a new species of Neopistolochopsis. So, the nurseries are trying to provide clean plants, of course, and they're trying to keep those out of their fields. And to do that, they basically rely on the same thing our growers do here in Florida or elsewhere, which is they use fungicides. And here's where the problem comes in. The nurseries basically have the same fungicides available to apply on their fields as we or our growers here have to apply in their fields. And at first glance, it might seem like there are many different options of products. But when you look at the fungicide groups and consider that you really can only use or should only use each one of those fungicide groups a few times, usually up to 4 times during the season, you can quickly see that the option for different groups is very limited. And that brings us to the issue that we are having more recently, which is the emergence of fungicide resistance to multiple fungicides and multiple pathogens. So I'm going to walk through some of those issues as a background, starting with Coleototricon acutatum. Back in 2013-14, we detected for the first time resistance in Coleototricon acutatum to the KOI fungicides. And you're going to see through my slides some of the students that have worked on those different projects. This is Bruna Forcellini here, and she was the one to report on the resistance to Coleototricon acutatum, which is due to a single point mutation on cytochrome B codon 143, and that resistance that mutation confers a complete resistance. Whether the isolates with this mutation, they are not controlled by any of the fungicides in that group. But to make things even more complicated, what we found is that resistance was actually coming already with transplants from the nurseries. So Coleototricon acutatum causes quiescent infections on plants, which means plants are infected you don't really see until you plant them in the field with the right conditions. Humidity in Florida usually is what it takes if the plants are infected. So what we found is that those plants were not only coming infected with Coleototricon acutatum, they're coming infected with strains that were already resistant to the KOI fungicides. And we sampled plants from 15 different nurseries. And out of the 15 nurseries that we sampled, we found resistance strains in 10 of those, which it makes really challenge for those growers when they're getting the plants. They don't even see their disease. They put them in the field. They realize they're infected, and the product that used to be the most important to control this disease is no longer effective. So it leaves them with very little options on that high investment that I just mentioned early on. So moving on to different pathogen, Phytophthora cactorum. So recently we have started picking up some resistance to mefenoxam. It's FRAC group 44. It's the commercial product is Ridomil, which is considered the most important and most effective product for control of Phytophthora in strawberries. So since 2016-17, we have been picking up some resistant isolates. The proportion in relation of the total and the population, it's still low, but very significant for the growers that get those plants that are infected. In this case, it's different than with coleotrichum in the sense that we can really pinpoint the resistance to two different nursery sources. And so those resistant isolates that we have here on the pie charts, they were all from the same two sources, which again, it's better for the growers, but not good for the growers that got plants from those two sources without knowing that they didn't have anything they could use to control Phytophthora on those plants. And then comes a botrytis, which I like to make this analogy as the MRSA of plant pathogens, because if resistance is going to show up, it's going to show up in botrytis first. So scientifically, our group has done really well publishing on all these resistant issues, but it's really a big challenge for our growers. And here, what you see on the pie charts, I have products from the different fungicide groups that I mentioned on the table earlier. And this slide is, in fact, from 2014-15. It's not up to date, but at that time, for all those 6 different groups that we tested isolates for, only one of those still had 100% of the population that's still sensitive. For all the other different groups, we had a good percentage of the population that was already resistant to those fungicides. And that was not only looking at populations in Florida, but we had a study where we collect isolates from several different states in eastern US. And the scenario was similar in all those different states, where pretty much everywhere we could find those isolates that are multi-site resistance, meaning that they're resistant to 4, 5, or 6 of those groups at the same time. So basically not much that will control those resistant isolates, similar to multi-drug resistance in humans. To our surprise, though, what we found that I didn't expect at that time is that botrytis resistant would also be coming from nurseries, because we don't think as botrytis is a problem that nurseries are trying to control, since they don't have fruit at their nurseries. Most of the time, they can have if they have the neutrals, but they're mostly trying to control coleotrichum and powder mildew. The problem is that they're using products from the same fungicide groups as the fungicide groups we have for botrytis. So Michelle is my student who did that work, and what we did, we collect isolates from the plants before they even went to the ground. So we collect plants from nurseries in California and in North Carolina, the different regions in Canada. And for most of those locations, we found the majority of the isolates already have resistance to 2, 3, or 4 fungicides coming with the transplants. Again, meaning there's only a few that will be effective in the fruit production field. So, and that's the background that has led to kind of change the direction of my program here in Florida, where I realized that I cannot only work for our growers here in Florida. I really need to come up with strategies that would also help those nurseries that are providing plants to Florida. So over the years, we have many projects that are have been funded with the overall goal of integrating disease management across nursery and fruit production fields for all those different diseases, with the idea that being an annual crop, we have a chance to reset every year. And if we can get clean plants to start with at the beginning of the season, we have much better chance to make a good crop and a good way to the end. So there are several different aspects that we've been working on. And of course, what I'll be talking today mostly is thermotherapy, which is a part of a broader objective where we are looking to develop some non-chemical management strategies in addition to other things. And heat is one of those strategies. So some history on heat treatment as a non-chemical alternative for management of diseases. It's not something new and not something I started. In fact, heat treatment had been used in nurseries and strawberry nurseries. And what I was able to gather back in the 90s, mostly for management of pests. And a lot of the nurseries were following a protocol to reduce cyclamen mites, it's a big concern. It was a big concern at the time. It's still a major concern, and there's not. It's difficult to control because the mites tend to hide inside the buds of the strawberry plants. So some nurseries were using the protocol that was based on this work from back in 91 from Bushner, and the protocol called for 7 minutes of treatment at 49 C for reduction of cyclamen mites. However, when I start working on these and I try to survey several nurseries, many of the nurseries had done at some point, but back off of doing this hot water treatment, reporting some issues with delayed growth, reduced flower. But in particular, what caught my attention as a plant pathologist, of course, is that they were reporting after they started doing this, they saw an increase in xanthomonas with this hot water treatment. So that's basically where we started. We wanted to see what would it take to kill xanthomonas in hot water. And I mentioned I've been working with Bill Turshak for several years on this. This work here in particular was published back in 2009, where we look at a temperature time combination that would take to kill xanthomonas. So here on the y-x, we're looking at survival proportion, and the x-axis, we're looking at time. So if you look at 49C for 7 minutes, we would be about here. And you can see at that temperature time, basically 100% of the bacteria survival. So the growers really had a point on that observation. If they're putting plants that have xanthomonas in a tank of water and they're not killing that bacteria, they end up spreading that bacteria and making it even worse. So looking at what would take to reduce the bacteria we found that it would take much longer exposure times to reduce the bacteria. But some of those temperatures that we look at, like 56 and 52, they're really too high for the plants. So we focus on those two here, 48 or 44 for 4 hours or 2 hours as a target for treating plants. So we did a series of trials with plants of different cultivars, looking at 44 for 4 hours and 48 for 2 hours. And sure enough, we had a really good control of angular leaf spot at those time temperature combinations for most of these cultivars. However, the issue we found is that hot water for a prolonged time, like 2 hours for 48, 4 hours to 44, also caused some plant damage. We saw some plant mortality for those cultivars compared to the control that was not treated. One thing we play with a little bit here, you see to have festival and festival dry. For festival dry, what we did is that we put the plants in sealed plastic bags. And before we put it in the water, kind of in a vacuum, and we found that the plants would take the heat better that way. However, it would be very labor intensive to bag the plants and assure the water would not get in before we put them in tanks of hot water. So in 2014, we were granted a SCRR grant that allow us to change our direction a little bit from what we were doing as far as heat with hot water. And the change was that we start looking at steam or aerated steam as an alternative to hot water. to heat treat those transplants. So we built a couple of prototypes to do the testing in Florida and California. And basically the one in Florida was a growth chamber that was not being used, that we adapt to incorporate some steamers on the side. The one in California was basically a truck container that we attached steamers. And this is wherein, who is an engineer from Norway and also a grower in Norway who helped us to build those chambers. And at that time and during the duration of that grant, we did a series of trials, few trials, looking at treatments for and let me back off a little bit. What we found during that time is that by adding a preheat step before we heat the plants for a prolonged time, we could activate heat shock proteins that would allow plants to take the 44 hours of heat for 2 or 4 hours better than we would do without the preheat. So we did a series of treatments looking at plants that were preheated at 37 for an hour, followed by the steam treatment, 44 for 2 hours, 44 for 4 hours, or treatments without the preheat. At that time, we kind of decided to back off on the 48 for 2 hours, mostly because we found that it was too borderline on damaging plants. It was effective on the pathogens, but it was a little more borderline on damaging the plants. So looking at specifically at pathogens, we knew at that point that the treatment of 44 for 4 hours would help to reduce Xanthomonas. So we wanted to see what this treatment would do for Polytrochon, which is another important problem that comes with transplants. So we basically looked at treatments with or without the preheat for 2 or 4 hours of duration, because it doesn't take as much to kill Polytrochon as it does for Xanthomonas. But what we found is that still 4 hours was better in reducing Polytrochon, of course, in addition to Xanthomonas. For the pathogen, it doesn't really matter if you have that preheat step or not as far as reducing the pathogen, but it does help for the plants. The plants take that hit better if you have that preheat step. So as I mentioned earlier, botrytis was another problem coming with the transplants. So we decided to look at botrytis as well. And in particular, whether we could reduce that population of resistant strains that are coming with transplants. So we inoculate plants with a mix of sensitive and resistant isolates, and we treated the same way with and without the preheat for 2 or 4 hours. And again, for botrytis, we found that 4 hours work better than the 2 hours. And like I said, the preheat helps with the plants. So that's the treatment or the protocol that we'll be talking about as we move on, that was developed from the SCR project. So with this CPPM project that was funded in 2019, and finally get into that, we basically wanted to expand and look at a more like what other pathogens and nematodes that we could control with this protocol that we had already developed. And we also want to expand a little further and looking at not only a plant survival, which we have looked at some previously, but also what effects the thermotherapy has on runner production if we're doing this in nurseries, or flower production, fruit yield, and a series of different cultivars. So starting with Phytophthora, we wanted to see what the protocol that would develop would do with Phytophthora cactorum. And what we found is that Phytophthora cactorum zoospores are actually highly sensitive to those temperatures. And in vitro here, of course, it only took 5 minutes to kill 100% of the Phytophthora zoospores. So it really didn't take much. Marcus and Juliana were the students working on that project at the time. However, Phytophthora produce different structures, as plant pathologists in the audience will know, and oospores tend to be more resistant to external conditions. So zoospores, I'm not showing any graphs because basically all the zoospores still germinate even at the 44 for 4 hours. So for our field trials, we decided to inoculate with both zoospore and oospore suspension to see if we could have an effect on both. The oospore, we actually only look in the second season when we realized that, that the oospores were not getting reduced in vitro. But basically in both cases and for both seasons, we saw a significant reduction in plant mortality when we treat the plants. And here again, we're only looking at the 4 hours with the pre-hit step. And for both, when we inoculate with zoospores and we inoculate with the oospores, we had a significant effect on reducing plant mortality caused by Phytophthora. With a Phytophthora, another effect of the disease is sometimes we see is that the plant might not be killed, but the growth is inhibited and the plants get stunted. So for the plants that did not die, we look at plant diameter. And what we found is that the plants that were treated, and whether inoculated with zoospores or oospores, they were significantly bigger than the plants that were not treated. And here on the side, we're only looking at the water controls. Sorry, I didn't mention that before. So good news for Phytophthora as well. The protocol that we have developed previously is effective against Phytophthora. And then we decided to also look at powder mildew, and we collaborated with Arne Stansveen from Norway on that one, where we basically look at a 4 for 4 hours. And in this case, we didn't even look at the pre-hit step. But what we found is that the 44 for 4 hours basically completely eradicate powder mildew. And we have to think powder mildew is pretty much an external pathogen. So it's much easier to kill than some of those other pathogens that are already infected the plant internally. So good news there as well. And for this grant, as I mentioned earlier, Johan de Seger is the nematologist here at our center. So he looked at the effect of heat treatment on 3 different nematodes: foliar nematode, Apheloncornis besseyi, lesion nematode, Proteolenchus penetrans, and root knot nematode, Melodoginum apla. And here is looking at the data for his in vitro trials. And in vitro, as you can see, for most of the nematodes, about an hour at 44 or higher, almost completely eliminate, except for here on lesion, it took a little bit longer to reduce nematodes. Here is, of course, looking at in vitro. When you look at inoculated plants, it does take a little longer because some of those nematodes are inside the tissue. So here we're looking at plants that he inoculated with those 3 different nematodes and treated at 44 for 1 hour, 2 hours, or 4 hours. And again, it does seem like the 4 hours works better in most cases than 2 hours, definitely better than 1 hour. So that's another pass that we add to our protocol that can be reduced or eliminated with the heat treatment. Here's some data on cyclamen mites, and that's where we started at the beginning with the history on the protocol for cyclamine and hot water. This was not even part of the CPPM grant, but I have collaborated with Justin Renkma in the past. And what he did is he basically treated it for only 44 hours because those mites are more exposed. It really doesn't take as long to kill the mites. So if you're treating a 44 basically for 1 hour, you can completely eliminate those hours. If you're doing the full 4 hours to take out the other pests and pathogens that I mentioned before, you definitely be controlling cyclamen mites as well. So as far as the effect on the plant, as I mentioned earlier, we had looked at extensively on plant mortality, but as part of the CPPM grant, we expanded to some newer cultivars. They're currently grown in Florida, like Brilliance and Sensation, as well as Radiance, that's highly susceptible to Phytophthora. And we didn't see much of a difference looking at control versus steam treated plants. And again, keep in mind here I'm talking about the 44 for 4 hours. We don't see any significant damage to the plants of that protocol. We did see some mortality here with Radiance. Radiance tends to be sometimes a weak, not very vigorous plant. But there's really no difference whether the plants were treated or not treated. We kind of had the same level of mortality. It was not really due to the heat treatment. As far as the effect on runner production, and that's a question we were getting from nurseries, is okay, well, the protocol can reduce all those pests and diseases. Will it affect runner production at the nursery? We have data from 2 years of trials with those different cultivars, which is still a limited set of cultivars compared to everything that's available out there. And what we found is that it seemed to be a bit cultivar dependent, where we saw some reduction in number of runners for Beauty and Brilliance, but not for the other ones. So that's an area that probably need to expand on the research as we're trying to treat different cultivars to see if we will affect the runner production. Nevertheless, with the discussion I had with nurseries is that they can deal with some reduction in runner production as long as they can produce some plants, and that those plants they're producing, they're clean or they have limited infection of pathogens. We also look at the effect of flowering. We never thought of thermotherapy as something as our growers here in Florida would do before they plant in their fields, and mostly because of the scale. We got 200 million plants in a matter of 3-4 weeks that we have to put it in the ground. So to treat that many plants in that short period of time, it wouldn't really be feasible. But I was getting some questions from some smaller nurseries that are getting plants from elsewhere that might have the ability to treat those plants. And the question was whether would it affect flowering and yield. So we did those trials with the same cultivars, looking at how many days it would take for the plants to flower, looking if it would slow down flower, because usually for most state, early production is where the growers make most of the money. And we really found a minimal effect of treating with the steam on the days that it took for those plants to flower. There are some exceptions here on Ventana, but so again, it might be cultivar dependent, but it is something that growers, smaller growers, if they can treat plants before they plant, that they could do to reduce the risks. And then finally, yield, we also found a minimal effect on yield. And in fact, some of the cultivars were even produced better when they were treated with the steam. So trying to wrap it up together, we really believe aridistin is the way to go for heat treatment. The advantages is that it has less adverse effects on plants than the hot water treatment. And we have now proven that it's effective at reducing a number of pathogens, including Xanthomonas, Chlorotoxicum botrytis, Phytophthora, powder mildew, as well as nematodes and mites. As the cons and something that we still need to figure out how to do it is that it does require some specialized units, especially if we're looking into do large scales. We need large units to be able to treat as many plants as those large nurseries have to treat it at a time. And it does take time. It takes about 6 hours for the full treatment because if we're considering the free heat step for 1 hour, and we usually give it an hour break to for the plants to activate those heat shock proteins, and then we follow up with the 44 hours with the 44 Celsius for 4 hours. We have published some details on how we build the prototype units that we have on the paper that I have here, in hopes that maybe something that some small growers or somebody that has some engineer skills could try to build their own units to treat the plants. And just going back to where we started, is the idea of the heat treatment is that we can reduce the spread of those diseases or those pathogens among nurseries. The way the nursery system works is that there's a lot of movement of plant material. Usually from California nurseries provide a lot of the plant stock to the Canadian nurseries and the North Carolina nurseries, but I have seen North Carolina nurseries providing plant stock to Canada. So there's a lot of movement of plant material. And if those plants going from California or North Carolina or elsewhere, if whatever they're infected with, that most of the times we can't see, they're going to be sent into those places. So we see as heat treatment as a way to reduce the risk of the risk of spread and the risk of epidemics. Because if we're doing that treatment every step on that propagation cycle, the idea is that we can keep that inoculum down, whether it's to the point where it's not going to reach epidemic levels. So just some final thoughts to wrap it up. We're doing good on time, still have time for questions and discussions. Is that I think all the data we have and that I have shown, I hope it convinces you to that heat treatment can be used as part of an integrated strategy to manage multiple diseases and fungicide resistance, in particular in nurseries. And it's not a single tool. We do need several different tools for integrated management, but the idea is that if we can incorporate some of those non-chemical alternatives, we can help to extend the life of those single-site materials and save them for fruit growers, and in particular for critical periods for fruit growers where the plants are in the ground already. They want to be able to use those products that are considered reduced risk, and those are usually reduced risk for as far as toxicity, but they're high risk for resistance. So we want to save those applications for the fruit growers, so they have to do only at a few key points during the season and not on a preventive basis like they've been doing in nurseries and sometimes in production fields. So with that, I'd like to thank my my group. It takes a lot of hands to collect all those data. This is a snapshot of group I have. Some of the many different groups I had over the years. I really appreciate every single person that has contributed to our work. And with that, I'll stop here, and I hope we have some time for some questions and discussion. Thank you.
Well, thank you so much. That was a great presentation and very thorough. And I want to remind people that are attending, if you have a question, feel free to put that into the Q&A box or raise your hand. I saw somebody raise their hand, and I will go ahead, Roger, and unmute you and let you ask your question.
Thank you, Kayla. Natalia, thank you so much for coming down and giving your seminar. It's really impressive to see everything that's going on, and I love the way you brought all these different people, including from Norway, together. I think that's awesome. A lot of different expertise. It's a great example of how to do a project. So firstly, congratulations. It seems like you've invented a sauna for strawberry plants. So I think that's really awesome. But my question is, like, how expensive is this equipment going to be to really to build? Is it something that like will be practical for a nursery to have one on site, or is it going to be something like more like there'll be one, treatment facility, say in California, that the nurseries could send their plants to before they get shipped to ship to Canada or wherever they're going next? So thank you.
That's a very good question, and nice to see you virtually. But it is a good question. I actually had a meeting yesterday with a company that produces plants in California and exports to other countries, and the questions and the discussion was whether they could treat those plants before they freeze them and ship them to those other countries. So we definitely don't have the questions answered, because I have never done the way they were asking, like, can I treat it and then freeze them? I always treat it and plant them. But their idea is that they would have a unit, they would treat it before they ship, rather than all those nurseries or growers have a small unit to treat plants as they receive them. I think there are going to be some different scenarios depending on the size of the nurseries. And in Europe, they're actually already starting to use this commercially. It's a different system that we have here, because they're mostly going in greenhouses. So actually the family of the engineer that worked with me in Norway, the son of Oli, decided to open a business and build sauna. So they have a commercial unit now that they're selling. To go back to the question on the cost, they're selling for, the last I heard, about $80,000 for a unit that can treat one pallet of plants, which I understand is about 60,000 plants. Which for Europe and greenhouse, to the scale that they're doing in the greenhouses, it's big enough. They are working on building a bigger one, which of course will be more expensive. But I started doing this thinking of nurseries, because as I said, I don't think our growers here can do it that scale. Nurseries, we're still dealing with lower numbers of plants. But I think there's room for different scenarios where a smaller nursery can build a smaller machine to treat plants that they receive from a bigger nursery before they put it in their fields or before they make their plugs. And there's room for big companies like Driscoll is actually, they've been working in California, and they have a couple of units, their facilities now, where they're treating their plants before they put it in the field. So I think we still have to figure out logistically how to make this work. And I don't think there's going to be only one way of doing, hopefully. I know I circle a lot, but I hopefully an answer.
Thanks. That was really great.
And we do have another question coming for you. This person would like to know if you've tried any remedies such as ice water or cold air after a heat treatment to reduce heat damage to the plants.
Yeah. So the way we do our trials here, the research center, is that because I mentioned the treatment takes 6 hours, and so we treat it during the day. At the end of the day, we put them in the cooler, and then we come back and we plant the next day. We have looked at like keeping them in the cooler for longer to see if it would affect their survivability. And we kept them in the cooler for as far as 10 days without seeing any effect on their survival. So yes, we do have to cool them after treatment to take that heat. The commercial unit they built in Norway, it actually automatically shuts down and starts cooling the plants once that 4-hour time kicks in. So, and again, trying to cool them down as quickly as possible to reduce that heat damage. But the damage, what we found, the damage with steam is basically not as much or as with hot water. So hopefully, if those plants get cool in a decent period of time, in a couple hours, there's no damage to them. I see another question here from Phil. Hi, Phil. For nurseries with known disease issues, could I start asking the nurseries to take on this responsibility? I'm thinking mainly of Neopistolocopsis at this point. Okay, so we have looked, and I didn't present Neopistolocopsis. We have looked at Neopistolocopsis last season, and we're planning to repeat this season. And Neopistolocopsis is a little beast. even in vitro, it took the whole 4 hours to completely inhibit, the whole 4 hours at 44 to completely inhibit Neopistolocopsis. So when we did it in with plants in the field, the 4 hours at 44, it significantly reduced the amount of disease, but it didn't completely eliminate. So I think it can help, but it's not going to be a silver bullet. I think it's going to be another layer of protection for Neopistolocopsis as well, but it doesn't seem to completely eliminate Neopistolocopsis.
Okay.
And the challenge, and just to add a little bit on some of those questions, one of the challenges, and I didn't go into those details and some of the trials we did it with nurseries, is to get that 4 hours at 44 when we try to scale up is challenging, because we often run into some cool spots, hot spots, and not all the plants were getting the full 44 for 4 hours. So if we're only getting the 44 for 2 hours, as you saw the data I show, we're not really getting the full benefit. So getting a uniform 44 for 4 hours on all those plants in larger scale is, I like to say, it's more an engineer challenge than a plant pathology challenge. Hopefully I'll have some good engineers to help us to figure out.
You definitely have to wear a lot of different hats when you're doing research like this.
Yep.
Well, Natalia, I'm not seeing any more questions, but I was wondering if you could leave the participants today with any kind of comment or just something for them to think about as they're leaving. So if you wanted them to remember one thing from your talk today, what would it be?
Ooh, okay.
Or a couple. It could be a couple.
Yeah, I think I already touched those things during my presentation, which I truly believe that we need. I often get questions from growers and nurseries and looking for a single solution, and I think we need to think of these and other things. And I didn't mention I've been looking at UV and it has to be an integrated approach where we are starting thinking what is the problem? Okay, what's my major issue? Is it Neopistolocopsis or is it Xanthomonas or what it is? And come up with an integrated approach for the major problems we're trying to deal with, rather than trying to look or rely on the silver bullet of, and often, of course, I think the fungicide applications. Fungicides are part of that strategy. We do need fungicides, but more and more we are losing them to resistance. We're losing them to the process of re-registration. Products that are not getting registered again. So, and companies are not coming up with new products as quickly as we're losing them. So we need to integrate those other approaches in order to manage all those issues, disease and fungicide-resistant issues.
Okay, great. I think that's wonderful and a great thing to keep in mind, because I feel like maybe the people on this call might know that to a certain extent, but it's always a good reminder. So thank you for that.
Mm-hmm.
And thank you again for your presentation today and for all the great work that you're doing. I did want to mention that we have a couple of other webinars coming up this month, which is abnormal, but we're really excited about them. So we have a webinar coming up on September 23rd on carpenter bead IPM. So if you want to find out more about integrated pest management with carpenter bees, you can check out that one. And then we have one a little bit later in the month on September 27th. And that one's going to be at 11 a.m. instead of 1 p.m. And that one's going to be all about how to get your grant proposal ready, if you're planning on submitting a grant proposal to us at the end of the month. So looking forward to both of those. Thanks again, Natalia, for your presentation. If you want to find out more information about the Southern IPM Center, you can visit our website, southernipm.org. And thanks so much for attending. Have a wonderful day.